Hi, I'm Nick, and this is Lab 1, Kinematic Design and Beam Alignment, my makeup. Here we have the 6 degrees of freedom. We have yaw, pitch, roll, X, Y, and Z. Once a mount has a 6 degrees of freedom constraint, then it is known as a kinematic mount. In this lab, we're going to align the laser. The laser is now mounted on a kinematic mount, which will help us align it perfectly. Here we change the tip tilt, which changes the yaw and the pitch. We also have a stage at the bottom, which allows us to change it in the X direction as well too. We also have a knob on the other side that allows us to change the elevation as well. In order to align the laser, we set up an aperture close to the laser and align the beam straight through the aperture hole. We then move the aperture down the rail and then proceeded to change the tip and tilt as well as the X and Y if necessary in order to align the beam through the aperture down the rail. We then verify by bringing the aperture back through close to the laser to verify that we have aligned the laser perfectly. After aligning the laser, we then place a pool of water orthogonal to the laser using a pentaprism to diverge the beam down to the pool of water and then back into the laser again. As we can see, the table is not set to gravity, therefore the two beams move separately as the table is shaped. If the table was set to gravity, therefore the beams would overlap and move in unison, the two beams being from the top surface and the bottom surface of the water. In this portion of the lab, we're aligning an optical element, which in this case happens to be a lens, to the optical axis. To do so, we adjust the tip and tilt of the optical element's mount. We then align the back reflection from the optical element to the laser, adjusting the X position and the Y position as well. This is to verify that the beam aligns with the center as well as the two dots from the first surface as well as the back surface of the lens. In this portion of the lab, we're setting up two lasers parallel to each other and using a pentaprism and a polarized beam splitter, because it was the only thing that we had in lab, to diverge the red laser perpendicular to the green and superimpose the red onto the green. After some alignment and an addition of a polarizer to the green laser, we were then able to get an orange dot indicating an alignment of both lasers. In this next section of the lab, we're going to align the microscope to the optical axis. We have the tip and tilt, which moves the crosshair reticle, which is at the focal point of the microscope's eyepiece and objective. To adjust and bring the spot back to the center, we adjust the X and Y of the microscope. By repeating this process until both the crosshair and the spot are centered, this will then align the microscope. We then move the microscope along the optical rail to verify that it is aligned again. To make this easy, place the dot on the wall after aligning the laser, and then aligning both the reticle and the spot to that dot. Another tip is that when aligning the polarizing beam splitter to get the orange dot is to use your hand to steady the mount while you take your picture. This is because the PBS is very sensitive due to its distance away. Also, another tip is to align the furthest optics first. This is to be able to move them closer to the source